Welcome to ICDD's InSession webinars. Anja Dosen, ICDD's Senior Scientific Editor, will present clay analysis.
Part 2.
Please remember to visit ICDD.com for information on future free webinars, educational events, product information, and to purchase PDF databases and Jade software.
Hi, everyone. Welcome to Clay Analysis Part 2. This time I'll be talking about some more details related to clay mineral analysis using X-ray diffraction and using our PDF database 5+ and Jade. So I'm going to go through some of the intro to clay that we've talked about last time, but I'm just gonna run through it just as a reminder, starting with the importance of clay. Clays are ubiquitous on our planet. They're essential as they contain nutrients for soil and for plant growth in soil. Clays can be used for remediation of pollutants. They are eco-friendly and cost-effective materials. They're generally used for wastewater treatments. We can use clay to design new materials with specific properties, like, for example, nanocomposites, which is also one big area of research. Of course, they're used in ceramic industry for porcelain, electroceramics, and so on. And an interesting point is that clays are also used in medicine and pharmaceuticals and biomedical fields, and they're used for drug delivery and also for tissue engineering. So all of these interesting facts are due to the clay having possibility of having expandable layers. So nutrients in soil are contained in these expandable layers. Remediation of pollutants, clay will absorb these pollutants in the interlayer space, and same thing goes for drug delivery. That is the path that drugs can be delivered. So a quick thing, what is clay? Clay is a naturally occurring material that's composed of fine-grained minerals. These fine-grained minerals are referred to as clay minerals. So what is a general explanation of clay? That it is generally plastic at appropriate water content, and it hardens when dried or fired. So that's, how we can make brick. So clay research is a very wide, broad field. It ranges from fundamental research that involves clay structural research and surface properties, all the way up to industrial and environmental applications and biomedical, as I mentioned. Clay minerals are layered structures. This is what makes them so interesting, is that they can have expandable structures. Clay layers are stacked along their basal planes. So what we can, how we can imagine that is like thinking about a deck of cards. So you can, you stack your cards, they will lay down flat on the larger surface area. That's exactly what clay minerals are like, and you can stack them in different ways, but they will always stack along the basal plane. They contain layers of tetrahedral sheets and octahedral sheets. Tetrahedral sheets are they're corner linked, and they have shared oxygens, whereas octahedral sheets will be coordinated with six or four oxygen atoms. So we have these, for example, clay layers. So we have a 1-to-1 clay minerals that are also called TO. So this is a tetrahedral sheet, and this is an octahedral sheet, and this here is the interlayer spacing. If we have 2-to-1 layers, then we have TOT. So tetrahedral, octahedral, tetrahedral, and then this here is the interlayer spacing. These interlayer space is filled generally with interlayer cations that will in the case of expandable clays, make the despacing appear larger. And in the case, and in some other cases, it can be hydrated, and then the despacing will appear even larger. So all of this, I talked about all of this in detail last time. So if you would like to know more or see more, all of our webinars are on our website. So you can go to ICDD.com InSession and take a look at our older or previous webinars. And I guess for now, Steph could also put a link in the chat box for you to have. So as I mentioned, clay minerals are platy. This is a really cool photo that I found. This is kaolinite. You can see how it's stacked. It has this hexagonal habit. And this is an SIM micrograph of kaolinite. So the bonding that I explained is what is responsible for the platy habit of clay minerals. Clay layers are bound together by the sheet of hydroxides. That is the interlayer spacing. So those are bound by ionic or hydrogen bonds. Or whereas the clay layers, they're bound together by covalent bonds. So as you can imagine, these are much stronger. So that sort of helps create this sandwich type structure, and that can also result in expandable layers. So if you think of, tetrahedral and octahedral sheets like 2 slices of bread, your interlayer cations or whatever is your, preferred sandwich filling, and depending on how much you like to put on your sandwich, it can be thicker or thinner. So that explains really well these like the expandable interlayer spacing in clays. So clays are very interesting, but also fairly difficult to characterize. If we're looking at a bulk sample, clay minerals always appear as oriented flakes, and their diffraction patterns are not necessarily really well defined. So when we have next to them a highly crystalline minerals or materials, we can lose clay peaks in the background. And what is a good practice is that we prepare clay samples separately. So we can do particle size separation, which I talked about last time, and do a separate XRD analysis of clay samples. So, but if we're looking at a bulk sample, we want to pay attention to broad and small peaks, low intensity peaks at low 2 theta angles. So besides, having preferred orientation and being sheet-like structures, clays are also interlayered very often. So we call this interlayering or interstratification, which means that clay layers are stacked along their basal planes, and we have these mixed layer structures that contain 2 or more clay types. So back to our playing cards analogy, if you take like 2 decks, a blue deck and a red deck, and you mix them all together and you take a look from a side, it's still going to be a deck. But then if you take a look, some cards are going to be from the blue deck and some from the red deck. So that's sort of, how we can visualize this clay interlayering as well, or stacking. So we can have ordered sequence where, stacking is done in some sort of like order, or a completely random sequence where, if we take 2 decks of cards and we just keep mixing them together. So when we look at the XRD patterns, we end up seeing an intermediate structure out of these mixed layer clay minerals. So here are some examples. This here is not a mixed layer clay mineral. This is clay. I just like this photo, and I think it was good to show you this. So here we can see a tetrahedral sheet. Then this green is the octahedral sheet, and then another tetrahedral sheet. So this is a TOT clay, And here we can see a wide gap, and this is the interlayer spacing that in this case is filled with water molecules. So when we stack clay, or when clays are stacked, we can have, so these here are some examples. So this is an AA clay. So this is one, pure clay, and these layers will be AAAA. And then we have a B clay, BBBB. So how, if these are stacked, they can be stacked in a perfect order, in which we call regular interstratification. So in this case, it would be A, B, A, B, A, B, or they can be stacked in a completely random way, and that is called a random interstratification. And we can't seem to find like any kind of order in this type of stacking. So as I mentioned, clay are gonna be always have preferred orientation, because they're gonna lay down flat on the longest on the widest plane. And we tend to analyze these oriented slides so we can analyze the basal spacing of clay minerals. Clay minerals are generally identified using X-ray diffraction of both oriented and random sample preparation. It is always best to analyze the clay fraction, which is less than 2 micron fraction. So I say here that oriented slides are easy to make, but last time we talked about the sample prep, and that is not an easy task. It is fairly time consuming. But once you're done with all that, it is easy to create an oriented slide. So if you were doing particle size separation, you would take out some of the suspension of less than 2 micron particles, and you can just drop it on your slide and let the clay particles float and orient themselves along the basal plane as they dry. And that way we can observe only the basal reflections from these clay minerals. There are some other specific treatments that we tend to do that help with characterization of clay minerals. I have here pre-treatment. Well, that's not something that we're gonna focus on this time, but there are some pre-treatments that can be done as well. And what we tend to do is saturate clays with some polar or organic molecule, like for example, ethylene glycol, which is most commonly used, because it's big and it will enter the interlayer spacing of expandable clays, and we will be able to visualize that on the X-ray diffraction pattern. Also, we perform cation saturation, and I'm going to be showing sodium and calcium saturated samples, which also influences the appearance of their X-ray diffraction pattern, and that also helps in characterization and clay determination. So talking about X-ray diffraction as a focus here. X-ray diffraction helps us in phase determination and structural determination. Each mineral or each material has its crystalline material has its own diffraction pattern. Those are like fingerprints, and looking at diffraction patterns, we can recognize the existence of disorder in certain phases or some phases that are disordered. We can see structural disorder, like some kind of structural defects, for example, and we can also observe interlayering using X-ray diffraction. Another interesting thing is seeing an amorphous phase using X-ray diffraction, which is this example here. Like these broad humps is what we call an amorphous hump. This is how we can determine the presence of an amorphous material. So I mentioned the disorder, interlayering, and all of that can be seen looking at raw data. So the importance of having raw data stored in the database has been recognized a long time ago. This here is an example of the same clay sample with raw data and without raw data. So this here is what we call a DI list, a list of D-spacing and intensities. And as you can see, it is much easier to visualize what this X-ray diffraction can look like if you have raw data present. So once you analyze your sample and you're looking at your own raw data, it is really good to have something else to be able to visually compare other than just the DI list. So the first raw data was published in 1979 in our database, and in that time that was paper product. It wasn't, there was no internet at the time. So this data was hand drawn, and here are the first PDF cards that contain raw data. And interestingly, they're all clay related. So here we see allophane, and allophane is amorphous or poorly crystalline clay mineraloid. So in this case, we can see it's not 100% amorphous, but it is very poorly crystalline or partially amorphous, as it has these two broad peaks. This here is the DI list. You see the list of D spacing and the list of intensities. Other than that, we had opal, which is very cool. We had 3 different raw data patterns drawn, which, as you can imagine, was incredibly useful. Having opal A as an amorphous opal CT as partially crystalline, and opal C as a crystalline phase. And another phase that had raw data was immobile, and that is also a clay mineral formed out of decomposition of volcanic ash. And here, for example, you can see D-spacing of 11.5, a very broad D-spacing. The intensity is 100%. That was drawn right here. These are 2 theta ranges. So this was in 1979. Today we have an online product. We have a very comprehensive database. This year, the ICDD came out with a new PDF5 Plus database, which has combined what was PDF4 Plus and PDF4 Organic. So now all the entries are within one database, PDF5 Plus, and this database contains over 1 million entries. And what is very cool is that we have almost 20,000 PDF entries with raw data. So within the PDF5 Plus database, we have different subfiles. And these are categories, mineral or material categories. So within the mineral related, we have a clay subfile. And as you can see, so far, we have 67 entries with raw diffraction data. We can display these. I'm going to show you this in a moment by mineral names and do different types of comparisons with our data. So PDF5 Plus clay subfile contains experimentally obtained clay XRD patterns, which is extremely valuable in different fields of clay mineral research. And I'd like to point out that the PDF5 Plus is a unique database that has such a feature. PDF5 Plus contains over 300 clay mineral entries, and those are both calculated entries that don't contain raw data because they were not analyzed, and experimental entries where some contain raw data. Besides raw data, we also have a comments tab in the PDF5 Plus that will tell you more about sample preparation and the conditions that the sample was subjected to. Why I'm mentioning this comments tab is because when you do a sample prep, even the best sample prep will not get rid of all of the impurities. So, for example, small amounts of quartz are always going to be present, and then probably feldspar as well. All of those can find their way into the the small size fraction, less than 2 micron fraction, and they're still gonna, in a way, interfere with your diffraction pattern. However, we have that noted. This is what I think is important. So in the comments tab, you will find which phases are present in these samples. This says kaolinite because this clay that we were looking at in this case, this was an illite-smectite mixture. Mixed layer mineral, not mixture, it's a mixed layer mineral, will contain kaolinite as well. So there was a kaolinite peak, meaning this peak belonged to a different clay. So you have all of these spacings that are listed for these other minerals, or in this case, impurities. We have this new project that I wanna talk about for a bit, and this is a project that is done. It's collaboration with Professor Stephen Hillier of the James Hutton Institute in Scotland. We have created a special grant for the submission of clay minerals for processing inclusion in the PDF database. And through this project, we're actively inserting more clay mineral patterns, which means that each time we have we have a new version of our database, we will have a more comprehensive clay mineral subset. Here is just an example of a PDF card. I'm gonna show you the database in a minute. This is an example of a PDF card of an illite that has raw data in the database. And as you can see, what I showed you earlier, it is very useful to have this, that you can visualize the peak shapes and not just the positions in our database. All in all, we have more than 300 clay mineral entries in the clay subfile. So I'm gonna switch and show you the database. So this is our search window. I'm gonna point here. As I mentioned, we have joined the databases, organic and PDF 4+. So you can search all data, but that search will take longer. Now that we know that we're looking at the clay subfile, we're gonna focus on inorganic data. Here are all the subfiles. You can see we have bioactive materials, cements, ceramics, of course, bioceramics, explosives, forensic materials, and so on. Here we're coming to metals and alloys, micro and mesoporous, which contain zeolites, and now we get to mineral related, which contain the clay subfile. So this search works really well if you wanna do a very specific search. I'm just gonna show you like a very simple thing. Like, for example, if you want to look at titanium oxide, you can say that you want to search only for titanium and oxygen. And if you perform this search, you will get all the titanium oxide that we have in our database. In this case, we want to search the clay subfile. So if I only choose clay here and don't change any other search parameters, and I click search, I will end up with 329 entries, which are labeled as clay mineral entries. So you can display various columns here, and what I have right now is a mineral name, because this is very useful when we're looking at clay minerals. You can look at the chemical formula, and I have these spacings, three strongest lines displayed right here. So, for example, if you have a sample and you're looking at a certain despacing and you're not sure what it is, you can arrange this by the largest D value and scroll through the list and see what matches yours, or several entries that might match your sample, and then start comparing them. What you can do, when I have this prepared here for you to see, we have, let me see. So here are two kaolinite entries. I have chosen entries with raw diffraction data. So the way, I'm just gonna back up for a second. The way I chose this is back to this screen. If I want to look at the entries that have only, that have raw data, I'm gonna click this here, and now instead of 300 and something, I'm going to get only 67 because this is what we have so far, entries with raw data. However, in the next PDF 5+ edition, we this number is going to go up to I think over 100. So the very cool thing about this is that you can observe the d-spacings, but you can also visualize the raw diffraction data pattern, and you can choose which ones you think could compare with your sample. So I have chosen these two kaolinites, and now we can compare the two PDF cards. What is cool when you're comparing, if you click through these tabs, they will change simultaneously on both cards. So once you get to comments, as I mentioned, you can see how these samples were prepared. Or what I mentioned about impurities here, this one starts, it says it has quartz as an impurity. Unavoidable, but it's good to know that you have it. And it says that this is a random specimen that's and then with this one, it will tell you that it's air dried and glycol saturated clay. So you can compare the difference between the oriented and unoriented clay, same clay, if you'd like. You can also, there's also a way of overlapping these two. So if you go and click on raw diffraction data here, and then you say add a diffraction pattern, I'm going to choose this PDF number and say add diffraction pattern. I want to add raw data, so here it is. You can still see, for example, this is interesting, basal reflections that are overlapping, and then you can see other reflections that we tend to avoid when we look at an oriented sample. So this clay mineral project, as I mentioned, we're coming up with new entries. We are adding currently 2 clay mineral standards, which are Wyoming from smectite. This is S Wyoming, and ICSZ. This is an illite smectite from Czech Republic. These are really interesting to have because they are very well defined. They have been defined by the Clay Mineral Society and clay mineralogists, and having these diffraction patterns collected under different conditions is very, very useful. So we're going to have 27 new entries that pertain to these two in the next edition of our database. We're going to have, as I mentioned, air dried, glycolated, heated, and saturated samples. So this is for just for your reference. I've shown this before. This is a quick just overview of how these spacings change once we treat the sample. So for example, we have smectite, sodium smectite. It has about 12 angstrom. I say about because it is never exact, depending on the humidity and different conditions. So we have about 12 angstrom despacing. Once we glycolate it, it will expand for like an extra 5 angstrom, and it will be a 17 angstrom despacing. If we dry it, and like if we heat it, not just dry it, it will go down to 10 and stay at 10. So kaolinite, for example, will get, the structure of kaolinite will get destroyed above 500 degrees. So I'm just going to show you some examples of these treatments, and starting with smectite from Wyoming. This is if you look at the major reflection, calcium and sodium saturated are going to have a slightly different despacing. Calcium saturated is going to have a larger despacing and a smaller 2 theta angle. So looking at the same basal reflection of smectite Wyoming, the difference between cation saturated and untreated sample is very distinct. Untreated samples has these broad peaks and lower intensity peaks, whereas cation saturated samples will show higher intensities and they will show sharper peaks. So if we look at the other two important basal reflections, we see again that the untreated sample has these very broad peaks, and then as we treated, the despacings change and the peaks become sharper. And here we have quartz peak, of course, and this is probably a feldspar. So what happens when we glycolate a sample? This is montmorillonite, that's a smectite, a very expandable clay. So if we look at, in this case, these are calcium saturated. If we look at air-dried sample, it shows a well-defined, fairly broad peak that if we when we glycolate, you see this shift. It moves from here to here. This is the same sample, only air-dried and glycolated, which means that the ethylene glycol molecules are in the interlayer spacings and the despacing is much larger, which we observe as a smaller 2 theta value. Same thing here, just even larger differences between glycolated and air-dried samples. So that was just smectite. Now I'm showing you a mixture of illite smectite. This is, again, I mentioned the clay mineral standard from, this is a 70-30 illite smectite interlayered clay. So if, what is interesting here, so you see, if you look at the randomly oriented sample, it shows a lot more reflections than oriented samples do, where we only observe the basal reflections. So this air-dried sample is showing an interlayered structure and it's showing an intermediate structure. Once we glycolate it, these are untreated, so it's just air-dried clay. And then once we glycolate it, you can see that we observe 2 peaks. This here is the smectite peak, which has absorbed ethylene glycol. And this is the illite peak, which hasn't, because it is not an expandable clay. When we proceed to heat these, they will go to a 10 angstrom peak. They will settle at that, as I showed in that slide earlier, and we can confirm that this here is a kaolinite peak because it is gone at 500, which means that it has decomposed. So here is a more detailed look at glycolated and untreated samples. You can see what happens here, how this one peak, I guess, splits into, or shows that we actually have 2 different types of layers in this clay mineral. What these samples look like if we treat them, for example, with calcium and sodium. In this case, calcium saturated sample would show slightly, slightly larger D value and slightly smaller 2 theta value. So we collected a local soil sample to show in this presentation. And this is done, we collected it from our colleagues' backyard. This is a soil sample collected in Swarthmore. This is a sample as is. As you can see, it's just, regular soil. So what we did is we dried it. We got rid of these little root branches and ground it a little bit and sieved it. And here is the sample that we analyzed once it was dry. You can observe like these little white flakes in the sample. So that's something, you know, that you can do a visual determination even before you do the X-ray diffraction analysis. These are micas. We live in the Philadelphia area. Rocks are metamorphic here. We're in the metamorphic core actually, and there is a lot of mica. So you know that by, you know, just seeing like all of these shiny rocks around. So we can expect to find this in our sample. So here is the diffraction pattern, and we start with the most obvious, which is quartz. We expect that. So yes, we're going to label this as quartz, and then we move on to other things, what we think may be present in this sample. So there's a lot of there are a lot of different clays. Kaolinite as a standard presence, then determine that this is actually muscovite, which is a mica, and not illite, because illite would be, you know, more of a clay mineral. Illite and muscovite are very, very similar, but in this case, this is really muscovite. So if we take a look at all the peak positions of all of these phases that we determined that we have in our sample, we can see that we have covered all of the peaks that we can see in the diffraction pattern. So clays, we have kaolinite, vermiculite, sepiolite, and dacite. So now we have to understand if we really do have these clay samples or not. So when we're looking at a diffraction pattern like this that has a very, very tall coarse peak and some other peaks, this is a different diffraction pattern where it was a little bit more difficult to determine the clay minerals. We want to focus on this area here. So up to 14, for example, degrees 2 theta. And as you can see, this is a very well processed pattern. This was done in PDF 5+, and I have removed the background so we can actually see this peak. But I'm showing this to you so you can see how small and broad this peak is, because before the background removal, you were not able to determine whether this is actually a peak or not. So this is something important to consider when you're doing clay mineral analysis. In our case, because this is soil, we have a lot of clay, and it wasn't so difficult to visualize it. So now we're looking at the same range of 2 theta up to 14 degrees, and we can see that all of the clays have been well defined. Justification is that all you have to do like some research. You can't just go and although the accuracy is very high, but you have to confirm that these are actually the clays that are possible to be in your sample. So for example, vermiculite is a commonly soil form clay. Sepiolite exists in Pennsylvania. So this is something that I was researching a little bit, because it is a semi-arid and arid regions clay, which is not the case where we live. However, it does occur in the Philadelphia region, so I was comfortable leaving it in as one of the clay's presence. And then dacite can occur in soils, and it is required to be determined using the X-ray diffraction, which we have done here. So now I'm going to move on and show you Jade. Jade is a very powerful tool from the ICDD and MDI. It is a comprehensive software that's used for diffraction analysis. What it does, it will help you do a phase ID. Jade works hand in hand with the PDF 5+ database. It will use all the PDF entries from the PDF 5+ database and help you determine your phase composition. And then also do further analysis like whole pattern fitting analysis, which will give you quantitative results of the phases you have in your sample. You'll get weight percentages, you can do ritual refinement, you can do clay mineral modeling, you can do clay peak determination based on a very handy tool that I'm going to show you. And these are just some of the features that Jade has. There are a lot more. So what I want to talk to you about is the oriented clays add-on. This is a very cool feature where you can model the clays. This is similar to NewMod. If you're familiar with NewMod, that's a program created by Robert Reynolds for the study of the interstratification of clay minerals. So this oriented clay add-on allows us to model the basal reflections of oriented clay samples. So we can create our models of interstratified clays. We can simulate mixed layer clays, and it uses the 18 what's called Reynolds structures, and we can modify those both structures and parameters. Okay, so now we're getting into something a bit abstract, but just bear with me. So how we model oriented clays. We do that using the layer transition probabilities. I was talking about layer stacking before. So we have the R value, Reichweite value that describes different types of ordering. So now the R value indicates, this is what's a bit abstract. The R value indicates the most distant layer affecting the probability of the occurrence of the final layer in an interstratified system. So, as I said, abstract. But really what that means is that we have R1, for example, that is going to be an ordered sequence. So if we have an illite smectite R1, we're going to have an IS sequence. So illite smectite. So the perfectly arranged R1 layers that are really, you know, one AB, or illite smectite will be deemed their own mineral. So for example, a perfectly ordered illite smectite is called rectorite. And it has an interstratified expandable, non-expandable layers. So ordering is dependent on the number of the interaction between the neighboring layer, meaning the one that's stacked on top. So we want to know how they stacked, and do they stacked in a way that permits X-ray data to predict this repetitive behavior, or to predict, you know, a peak shape. That's what we end up getting. If we have an R0 that is a completely random sequence, so there is, we don't know, there's no interaction, they're just super randomly stacked. If we have R1 sequence, and I said that's an AB sequence, or illite smectite or whatever, then we end up seeing an intermediate structure on the X-ray diffraction pattern. So we can say that the proportions of A and B are 1, meaning 100%, or the probabilities. So if we have a perfectly ordered sequence, then the probability of layer A followed by a layer B is 1. And the probability of layer A followed by a layer A is 0, meaning A can never follow A. However, if we have like a 70-30 mixture, as I mentioned, then we have more B than A, which means that A cannot follow A, but B can follow B. So we can have higher stacks of B, and A will always be followed by B. So it follows that kind of logic. It is a little bit abstract, but it follows a logic. So when we go to the oriented clays, which I'm going to show in a minute, we can do phase matching. We can, first of all, we can do phase modeling. We can model our own phases and create our own database of model phases. We can do phase matching and let the software do that for you. And then Once it does that, depending on the complexity, it can go up to like searching thousands of patterns to simulate to compare to your sample. And then there's a pattern mixer that will show how your chosen clay minerals are mixing in which amounts, and you can change those parameters as well. And you can run the software to generate the mixture. Okay, so now I'm going to show you this add-on. So this is Jade. I already have a clay pattern loaded. This is an illite smectite oriented, untreated, air-dried sample. We already looked at this one earlier. So first, what I want to show you here, you have this clay despacing. So clay despacing, what it does, you can put your cursor on the peak and it will give you suggestions. It will say possible clay minerals in cursor vicinity. It gives you an error, or not an error, an area that's looking in. And it will say this is the peak of smectites in various hydration states, which is true. If we move on to this one, for example, which I see what it does if I select this peak correctly, it will say rectorite. And I've mentioned earlier that rectorite is a perfectly ordered illite smectite mixture. So this peak is represented of an R1 illite smectite mixture, and that is rectorite. So now I'm going to show you the oriented clays. So what it did, it has already loaded our sample. Our sample is the measured sample. It's this magenta color here. And what I have here, is just kaolinite selected. So it's just one clay. And what it does, it's showing me that this peak here is kaolinite, just confirming it. I want to unclick single clay. And now I have two clays that I can choose as mixtures. So you can model this now. We know what this clay is, but let's say that we can do trichlorite, and then the second clay is going to be smectite, and it's going to show you the basal peaks of this mixture here. So in our case, we know that our first clay is going to be dimica or illite, and then the second clay is actually going to be this one. It's going to be dismectite with 2 waters, or maybe dismectite with 1 water. So now we have this peak better. We can go on through this. It's not going to be glycolated. It is going to be with water, trismectite with 2 waters. So we can choose whatever best fits us here. So let's go with dismectite and 2 waters. So, I mean, you can model it here. You can change your despacing here as well, and you can see how it's being modeled. So as I said, you can mess with all of these parameters. You can change the ions. You can go here and show overlays. You can show which clays you have. It will show you different despacing, different lines from the clays that you've chosen here. And if we go to tools and we click phase matching, I'm going to get this box, and I'm going to choose here which phases I want to look for. So in our case, I'm just going to stick to micas and smectites. I'm not going to choose anything else. But I mean, once you work with this, you can play with it and see how it goes. And if you have an unknown clay, you can choose a lot more than this. So once I run it, it's going to go through, in my case, hundreds of patterns, simulations, really. in some other case, it can go up to 2000. So now it went to, I think, like 300-ish. So we can, once you start clicking through this list, you're gonna see how these chosen clays compare to yours. So I'm gonna say this one, and I'm gonna find kaolinite here. And I'm gonna choose just those two. So now what I can do is I can go here and say create a mixer set, and it will create these mixed patterns, and I can do some further analysis from here, but it's right now it's giving me an estimate of the weight percent of these phases based on the diffraction pattern. So now, if I go back here and I right click and say create GA set, I can do this through tools tab as well. This is something that's called a genetic algorithm. This is going to create different simulations of these clays and determine all of these parameters that are weight parameters, proportions. This is ordering, preferred orientation, background, and so on. So if I click on this, you'll see it is starting with random sequence, and this is the generation 1. This is done so that it resembles a or organic analysis. This is sequence like chromosomes, so it will move on to the new phase of like reproduction, crossover, and mutation, where crossover would be the mixing of phases, mutation would be a random change. It would randomly change a parameter or a bit, and so on. So once you, again, it's looks a little abstract, but it is very useful. So you can, as it goes through the generations, it will model your diffraction pattern, and you can observe that here. So I'm gonna stop this and show you the final product, because we don't have that much time right now. So here is our simulated pattern after several cycles of this analysis. So we don't see all the parameters here, but we can see that it went up to, it has a really good R value. This is very questionable, but this is because I scaled it, and I'm pretty sure that it's because of that, but I have to double check. But you can see that it is doing a really good job in simulating your pattern. So I just have, okay, so then you can also, this screen shows that you can, when you're choosing your clays, this is when you're doing your own modeling, you have an option of seeing the chemistry of these clays, because as I mentioned, these are model clays. And you can look at their chemistry and understand if it makes sense related to your sample or not. So, okay, after all of this, I would like to promote a little bit the Clay Mineral Society. We've realized these clay talks have, there's a lot of interest for these clay talks. And I'd like to point out that there's a lot of, there are a lot of benefits to Clay Mineral Society membership. So membership, the biggest thing, I guess, is that membership is free for students and early stage researchers, early career researchers. And there are a lot of benefits of being the Clay Mineral Society member, like, you know, networking, of course. There are a lot of educational options, webinars, talks, and so on. Also, cool thing, travel and research grants for students. And there are all these different discounts that you get as the member. And I guess I'm gonna leave you with a whole new area for clay research, which is Mars. This is the Opportunity rover took the photo on Mars, and we already know that there are clay minerals. So as I said, a whole new research path for clay mineralogists. Thank you.